polyaniline on the removal of methylene blue, an industrial dye. So polyaniline-based polymers act as an efficient adsorbent for the removal of toxic industrial dye from water. Here we comparing the doped and dedoped form of polyaniline, and for the okay, so here we compare the doped and dedoped form of polyaniline to understand which structure is better for the dye removal and why. So here we are not only focusing on the dye removal, but also the role of polymer modification.
Okay.
So there's a large number of wide variety of textile and chemical industries worldwide. These industries use over 10,000 different dyes and pigments. These dyes are very stable and very difficult to degrade. These industries discharge a large amount of colored wastewater into the environment, which causes several environmental pollutions. So methylene blue. Here we are discussing about methylene blue. Methylene blue is one of the most commonly used dye. It is a cationic dye, that is a positively charged dye used in textile industries, and it also has some biological applications. So here the structure of methylene blue is here. It has an aromatic planar structure, so it can easily delocate into the structure of DNA and can break its strands. Methylene blue is carcinogenic and also has mutagenic properties. So it is really harmful to humans and other organisms. When this methylene blue reaches water bodies or water, it colors the water and prevents the sunlight or light which falls on it to penetrate to the deep layers of the water. This causes the depletion of dissolved oxygen in the water, which causes the death of organisms and the ecosystem. So removal of methylene blue is environmentally really important. Okay, so I already said that methylene blue is a cationic, that is a positively charged dye. So it can easily interact with negatively charged conducting polymers like polyaniline, and which allows the process of adsorption. So what is conducting polymers? We already know that polymers are made from simple molecular units called monomers through polymerization. But not all polymers can conduct electricity. Conductive polymers are simply biological organic polymers. They can conduct electricity due to the presence of pi conjugated system along its backbone. So these pi electrons can delocalize and can move along the bond. Here it's conducting polymers are classified into intrinsic conducting polymers and extrinsic conducting polymers. So intrinsic conducting polymers means it has its own conjugated structure. Extrinsic conducting polymers, we need to make it conductive by adding another conductive element or blend it with any other conducting material. So the conductivity of a conducting polymer can be increased by doping. So here for the removal of methylene blue, we take polyaniline as the conducting polymer. So polyaniline is a conducting polymer which is derived from aniline. It exists in different oxidation states. The three major oxidation states are for emeraldine salt, emeraldine base, and pernigralin base. It can we can dope polyaniline using acids as well as other dopants. So the preparation of polyaniline, we take a 4.65 gram. We take distilled aniline and HCl solution in a beaker. To it, we take ammonium persulfate solution in dissolved in HCl in a burette and added dropwise to the aniline solution, and we with constant stirring, and we after a 1 hour we get a green colored precipitate which is polyaniline. We let it dry and filtered it and powdered it. So that's how we obtain this polyaniline. And doping and doping of polyaniline, doping can be done by using acids like hydrochloric acid and H2SO4. Here we use the HCl. So what really happening when we doping polyaniline was the imine nitrogen of the polyaniline get protonized by the acid, and we can depol and we can dedope it using a base like NS4OH. So adsorption. So adsorption is the process of attachment. Molecules get attached on the surface of a solid. Here, what happened was the methylene blue, which is a positively charged methylene blue, get adsorbed on the negatively charged polyaniline through electrostatic interaction, and also there is a pi-pi interaction between the aromatic rings of methylene blue and polyaniline. So for the method, we take aniline as the monomer and ammonium persulfate as the oxidant, and here we use HCl as the dopant. So the emeraldine salt was first synthesized using HCl, and we dedoped it using NH4OH. And we get both doped and dedoped form of polyaniline. These doped and dedoped polyaniline mixed with the methylene blue solution and stirred for 1 hour. After the filtration, we determine the adsorption adsorption of the resulting solution using colorimetry. So on the we look at the results, we can see that the doped polyaniline exhibit enhanced the adsorption of methylene blue compared to the dedoped polyaniline. This is because the doping increases the surface area for adsorption and the functional group, the availability of functional group, which is favors adsorption after the doping. We take X-ray diffraction measurements for the to study structure of polyaniline. Here we can see that the polyaniline has an amorphous nature of broad peaks, and there is no sharp peaks. But doped polyaniline, we can see these sharp peaks, which indicates the improved crystallinity due to the polymer arrangement, alignment of the polymer due to are agglomerated with a size of approximately 100 nanometer, and it shows a block and spherical structure. And we also take TGA and particle size analysis. Here on TGA, at 50 to 110 degrees Celsius, the release of moisture is occurring, and then the dopant removal is occurring at 115 to 240 degrees Celsius, and the oxidative degradation occurs at the temperature range of 340 to 600 degrees Celsius. And by analyzing the particle, we get that the size of the particle found to be approximately 0.1 micron, which is equal to 100 nanometer, which is already we get it from the SEM analysis. And the further scope of this study is to found another different dopants which can improve the performance of polyaniline, and can also do other characterization, advanced characterization techniques for the study the adsorption of and morphology of the polyaniline, and can also regeneration and reusability of polyaniline can be also. Right. So by conclusion, we got that this experiment, we get that doped polyaniline shows enhanced the doped polyaniline exhibit enhanced the adsorption of methylene blue than the dedoped polyaniline. This is because the doping enhances the interaction sites and the availability of functional group, which favors the adsorption. And PANI has extensive applications. So PANI can be used for the removal of wastewater, and also due to its conductivity, we can use it in batteries and sensors, etc. And thank you.
Anusri, you have prepared polyaniline from aniline. Okay. Then in that scheme, what is the role of ammonium persulfate?
It is used as an oxidant.
Oxidant.
to
Then-
enhance polymerization and oxidation.
Okay. Then how did you confirm the formation of polyaniline? Is there any, have you done any specific techniques to confirm the formation of polyaniline from aniline?
Okay, when we obtain the dark green colored polyaniline,
Uh,
uh,
Here the monomer is aniline. You polymerized it and you
Yes.
got polyaniline.
polyaniline.
And did you check the molecular weight of that compound?
Yes, we then checked it. And
How?
മാം ദിസ് ഫസ്റ്റ് ഓഫ് ഓൾ ഞങ്ങൾ സീനിയേഴ്സ് ചെയ്ത് വെച്ചതായിരുന്നു
Okay.
പോളിയനിലിൻ വരെ. അതിന് ശേഷമുള്ളതായിരുന്നു നമ്മൾ ചെയ്തിട്ടുണ്ടായിരുന്നത്. സോ ദ മെത്തേഡ് ഈസ് ഓൾറെഡി അവർ ചെയ്ത് പോളിയനിൽ പ്രിപ്പയർഡ് ചെയ്ത് വെച്ചതെന്നാണ് നമ്മൾ സ്റ്റാർട്ട് ചെയ്യുന്നത്. ഓക്കെ. HCl acid. നമുക്ക് ഈസിലി അവൈലബിൾ ആയിട്ടുള്ള ആസിഡ്സ് H2SO4 HCl യൂസ് ചെയ്യാം എന്നുള്ളതായിരുന്നു. സോ H+ പ്രൊവൈഡ് ചെയ്യുന്ന സമയത്ത് ആ പോളിയനിലിന്റെ പ്രോട്ടോണേഷൻ അത് ഒന്നുകൂടി ഹെൽപ്പ് ചെയ്യും, എൻഹാൻസ് ചെയ്യുന്നുള്ളത് കൊണ്ട് HCl HCl.
നല്ല വർക്കാണ് കേട്ടോ. അതിലെ മെത്തിലിൻ ബ്ലൂവിന്റെ കംപ്ലീറ്റ് ഡീഗ്രഡേഷൻ നടന്നിരുന്നോ?
കംപ്ലീറ്റ് ഡീഗ്രഡേഷൻ മീൻസ് ആ TG അനാലിസിസ് അത് മാത്രമേ നമ്മൾ ചെയ്തിട്ടുണ്ടായിരുന്നുള്ളൂ.
അല്ല, ജസ്റ്റ് ബൈ വിഷ്വൽ, ബിക്കോസ് ഐ ഹവ് ഡൺ ഇൻ ദിസ്. അതുകൊണ്ടാണ് ചോദിച്ചത്. കഴിഞ്ഞ പ്രാവശ്യം നമ്മുടെ UG പിള്ളേർ ചെയ്തിട്ടുള്ള ഒരു വർക്കും കൂടിയാണ്. അതുകൊണ്ടാണ് വി ഹാവ് ഡൺ ഇൻ അനദർ വേ. അതുകൊണ്ടാണ് ചോദിക്കുന്നത്. മെത്തിലിൻ ബ്ലൂ ഇൻ കളർ, റൈറ്റ്? Okay, if it is undergoing any degradation, complete ആയിട്ട് വൈറ്റ് ആയിട്ടോ കളർലെസ് ആയിട്ടോ അങ്ങനെ മാറിയോ?
കംപ്ലീറ്റ്ലി എന്ന് പറയാൻ പറ്റില്ല.
ഓക്കെ. അപ്പൊ നിങ്ങൾ യു ഹാവ് കമ്പയർഡ് ദ സ്റ്റഡി വിത്ത് ഡോപ്ഡ് ആൻഡ് ഡീഡോപ്ഡ്. ഓക്കെ, ഇതൊന്നുമില്ലാതെ വെറുതെ മെത്തിലിൻ ബ്ലൂ എടുത്തിട്ട് ചെയ്ത് നോക്കിയോ? എല്ലാ പാനി ഇൻ സിമ്പിൾ മെത്തിലിൻ ബ്ലൂ വെച്ചാൽ ഡീഡോപ്ഡ് പാനി അല്ലാതെ വെറുതെ പാനി എടുത്ത് മെത്തിലിൻ ബ്ലൂ ആയിട്ട് ബ്ലാങ്ക് റൺ ചെയ്താ?
Actually, I'm doing doped and polyaniline ഞാൻ ചെയ്തിട്ടുണ്ട്, doped and deep-doped. So
You're still doing it's
another paper.
Okay. ഇത് കംപ്ലീറ്റ് ചെയ്ത വർക്ക്. ഓക്കെ.
ചെയ്തതായിരുന്നു. സോ അങ്ങനെ ചെയ്തിട്ടുണ്ടായിരുന്നില്ല.
ജസ്റ്റ് ചോദിച്ചു എന്നേ ഉള്ളൂ. ബ്ലാങ്ക് റൺ ചെയ്യണ പോലെ റൺ ചെയ്തോ, ജസ്റ്റ് ഔട്ട് ഓഫ് ക്യൂരിയോസിറ്റി. നല്ല വർക്കാണ് കേട്ടോ.
So next paper is presented by Sneha MB from Kerala Varma College, Thrissur. Next paper is presented by Shalima from Kerala Varma College, Thrissur.
Good afternoon. My name is Shalima. I'm from Kerala Varma College, Thrissur. Now I'm introducing the paper Synthesis, Spectral and Biological Study of the Nickel Complex of 3-ethoxy-salicylaldehyde benzoyl hydrazone. This shows the condensation of hydrazone. We know that hydrazones are the compound obtained by the condensation of carbonyl compound and a benzoic hydride part. The introduction of the CO group to the hydride part increased the electron delocalization and the density. Hydrazone has wide range of applications such as anti-cancer activity, antibacterial activity, antifungal activity, antiviral activity, anti-inflammatory activity, etc. All of this. So we are considering the synthesis of aryl hydrazone. The synthesis is already reported. So it is prepared by the mixing 3-ethoxy-salicylaldehyde and benzoic hydrazide in 1:1 ratio. Dissolve the DMF and methanol, and the two solutions are mixed and refluxed for 4 hours in the presence of acetic acid. From that, we get the aryl hydrazone. From that aryl hydrazone compound, we are making two metal complex In first complex is prepared by dissolving the hydrazone compound in the nickel methanolic solution of the nickel acetate, and the resulting solution is refluxed for 3 hours. So we get a yellow compound of the first compound. The next nickel complex is prepared by the same method. The hydrazone is dissolved in nickel acetate, methanolic solution of nickel acetate, and refluxed for 3 hours. After 1 hour, 2,2- bipyridyl is added and refluxed for 1 hour. And this complex structural studies are done by elemental analysis, IR and UV. In elemental analysis, we expect a structure for these compounds, and it is drawn in the chemdron. From that, we get the theoretical values of the percentage of carbon, nitrogen, hydrogen. So if we given for it analysis, from that we get the experimental value. The difference between the theoretical and experimental value must be in the range of plus or minus 0.6 value. If the range is correct, we expected structure is correct, and there is also a magnetic moment. This is due to the presence of unpaired electron. It is measured by Gauss balance. So we get the effective magnetic moment. From the IR studies, this is the spectra of 3 ligand and the 2 metal complex. From that, we can say that in the ligand, there is a presence of CO and NH group, which show the 2 peaks. But when it's come to the complex, the 2 peaks are disappeared because during complex formation, it converts to enol form. So the CO group is converted to COOH, and the NH group is converted to N double bond N. That is why the peaks are disappeared. It is the conformation of the formation of complex. From the UV spectral analysis, there is a intra-ligand transitions are observed in the ligand. If we come to the complex, the shift range is slightly changed. In complex, there is also in charge transitions are also observed, but here the DD transitions are not observed because these ligand charge transfer are more stronger than DD transition, which masks the DD transition. It finds an application in vitro cytotoxicity studies. Cytotoxicity means the death of the cells, and vitro, the experiment is done in animals. So the tumor cells washed with PBS or normal saline, viable cell suspension added to tubes containing various concentrations of the test compound, and the volume was made up to 1 ml using phosphate buffered saline, incubated for 3 hours at 37 degrees Celsius. Cell suspension was mixed with 0.1 ml of 1% tryptophan blue and kept for 2 to 3 minutes and loaded on a hemocytometer. Dead cells take up the blue color of tryptophan blue, while the leaf cells do not take up the dye. The number of stained and unstained cells were counted separately. And the percentage of cell death is obtained by the number of dead cells divided by the number of leaf cells plus number of dead cells into 100. This table shows the concentration of the drug and the activity of the ligand as well as the metal complex. As the concentration increases, we can see that the activity of the metal complex as well as the ligand increases. From this, we draw a graph, percentage of cell death versus the drug concentration. We can see that as the concentration of the drug increases, activities also increases, but the activity of the metal complex is more than the activity of the ligand. So we can summary and conclusion. 2 nickel complexes were synthesized and characterized by various physiochemical techniques. Metal complexes are penta and tetra coordinated. From the elemental analysis, we can get that in vitro cytotoxicity studies of aryl hydrazone and its complexes were carried out. Metal complexes are more potent than aryl hydrazone because the complex, metal complex has a planar flat aromatic structure, and the hydrazone moiety has that planar structure, and it has more hydrophobic in nature. That is why metal complexes are more activity in cytotoxicity. Yes.
Then what is the yield of this product? Yield.
ഇത് മാം ഇത് കംപ്ലീറ്റ് ആയിട്ടില്ല,
എത്ര കിട്ടി yield?
അത് ചെയ്ത് നോക്കിയിട്ട്
ചെയ്ത്
അറിയില്ല.
നോക്കിയിട്ടില്ല. Okay. ഏത് ആപ്ലിക്കേഷന് വേണ്ടി
ഇത് ആന്റി ക്യാൻസർ ഡ്രഗ്.
ആന്റി ക്യാൻസർ ഡ്രഗ്.
The next paper is presented by Sneha MB from Kerala Varma College, Thrissur.
As per the presentations are presenting here, my topic is self-folding behavior of polyaniline in but just a drop of water. So we can make it useful in soft robotics and medicinal purposes and drug delivery and etc. So that's the main purpose of our study. So these are some nature stimuli responsive functions. So the first one is venous flytrap. we already know that what the purpose is. When it is triggered by a prey or something, it will shut his two lobes of the leaves. It's a defensive mechanism or adaptive mechanism, nature-based engineering mechanism. And the second one is a pine cone. To protection of its seeds, when it absorbs water, it becomes shrink. And when it dry, it comes into an original state. So it is also an adaptive mechanism. And the third one is we call a touch me not. This plant, when we touch it, it will start to twirl inside its leaves. So it's also an adaptive mechanism. And there is also some man-made stimuli responsives. These are some man-made stimuli responsives also. The soft gripper and cell capture. And these are the sheets of polystyrene and coated with light absorbing substance. So when the light comes, it will start to fold. This also some man-made stimuli responsive. I just showed for the reference. So by inspired by this all, that's the topic what we are entering. The incorporation of polyaniline with chitosan with the response of water. So as per the first title, the first material is chitosan. The chitosan is a potential polymer. It as per the structure, it has primary functional group has amino and hydroxyl groups. It's highly easily for hydrogen bonding. And chitosan itself is not that much as stable, that much as brittle. It's highly weak. So we can modify it. Here we modified it with glutaraldehyde. So it becomes linked polymer. And its promising features are biocompatible, biodegradable, inexpensive, non-toxic, antibacterial effect, film forming ability. And as I mentioned before, it has low mechanical properties. And so our title is the self-folding behavior. So what happened with chitosan itself? So the rose-colored film is a thin film of chitosan. And when it placed in above the water, then it started to absorb the water and it started to fold. And when it completely absorbs the water, it started to defold. The mechanism is, I earlier mentioned that the chitosan film has amino groups and hydroxyl groups in the structure. So the amino groups and hydroxyl groups are interact via hydrogen bonds. So when the water comes, the hydroxyl groups forms hydrogen bond with the water. So it can easily diffuse through the chitosan film. That's why it leads to the folding. There forms compression and stress that started to relax. That's why it started to defold. So that's the purpose. And it's only at the time of the folding time is only with 35 seconds. And can be irreversible. It's reversible in nature. It started to defold again. So if we wanted to make it irreversible, we can use it rasosinol with incorporation, but that's not as per the study of ours. We are just mentioning that. So this is an experimental setup that we recorded the video of the folding. This is a goniometer and the recorded was, this is the folding. When the water absorbed, it started to fold, and after a few seconds, it started to defold. And its application we can use in soft robotics, artificial muscles, optical devices, and sensors, and also. So the next compound we use is polyaniline. Polyaniline is a first non-black aniline. It can be in different form with doping and oxidation level and etc. So it has its high environmental stability and its simplicity. So the preparation of polyaniline is, we weigh a definite amount of aniline, distilled aniline in 500 ml beaker and add 100 ml of HCl. And on another side, we add ammonium perfate in 100 ml HCl and take drop into the aniline solution and stir the mixture for 1 hour, and we got green colored precipitate. And stand for 1 day and filter and dry the polyaniline, and then it's powdered. So the synthesis of polyaniline, we put a definite amount chitosan in water and acetic acid, stir it for 4 hours with a magnetic stirrer under 35 degrees Celsius, add 3% of polyaniline that we prepared earlier, powder into the chitosan solution, stir another 1 hour for uniform mixing, add 300 ml of glutaraldehyde, pour into petri dish immediately to form the film. This is what we got. So we characterized polyaniline. The first characterization method we done is thermogravimetric analysis, and the next one is dynamic light scattering. From thermogravimetric analysis, we analyzed that there's 3 decomposition. The first one is the release of moisture, and the second one is release of dopant molecule, and the third one is oxidation of polymer chain. So the release of moisture happening at 50 to 110 degrees Celsius, and molecule release actually happen at 115 to 240 degrees Celsius, and then the oxidative degradation. That's why we call it a stable. And particle size is approximately 0.1 microns. So initially, I mentioned that the folding behavior of chitosan itself. Here, what happened with the incorporation of polyaniline into it? So when polyaniline added to the chitosan, it becomes started the permanent folding. That is, firstly, it has 35 seconds for the folding. And when we added polyaniline, it become reduced to 27 seconds. And it also increases the permanent irreversibility nature. So the basic mechanism is that when a polyanilyl presence is there, hydroxyl groups are increased. So hydrogen bonds are increased. So the attraction are increased. So the folding property also increased, and then the time of folding is decreased. So the rate of folding and irreversible nature increases with the polyaniline or chitosan film. So we done SEM. For the SEM images, we analyzed that the size of the molecule is 100 nanometer. That we also got from DLS. And there is more spherical and block-shaped particles from the morphology. So this is what we done on the experiment. We cut the chitosan incorporated polyaniline sheet, the film, into a flower shape, and then we put thermocol in the central side for the weighting purpose, because I also that low mechanical support. So when we apply the water downside, then it started to fold. That's what we are observing here. So the conclusion of our topic is to determine the suitability of chitosan films to undergo folding and unfolding process. Then chitosan itself, it takes 35 seconds, but with incorporation of polyaniline, it takes only 27 seconds. So we can make it irreversible in nature. And I think for the purpose of it only triggered by the wet molecules, then we can useful in soft robotics. So that's all about our study. Thank you.
Is there any evidence for the presence of bonds between polyaniline and chitosan? Any characterization you have done? Okay, so you have to do it, whether it is just a mixture or how the properties of chitosan that is affected by the addition of polyaniline. Then what is the purpose of thermal stability? You have done TGA. TGA. This is whether you are using in water, then why you are taking thermal stability? Any hot temperature application is not using. We are doing it in water. Then what is the purpose or relevance of this TGA? Okay.
Okay. Is it possible to adjust the folding time? Okay. Okay. All time you got 30 seconds. No, all times. Okay. Then you have prepared this composite by solvent casting method, na? Okay. Preparation method. I'm asking about the preparation of this composite. Polyaniline chitosan composite. How you prepared that composite? Okay. Then how you mix these two components? Did you use any solvent? Okay.
ഫിലിമാണോ ശരിക്കും പറഞ്ഞത്? ഫിലിമായിട്ട് ഈ കോമ്പോസിറ്റ് തന്നെ ഫിലിമാക്കിയിട്ടുണ്ടോ? അങ്ങനെ ഫിലിമാക്കിയിട്ട് അതിനെ മോൾഡ് ചെയ്തോ?
ഫിലിം മാറ്റിയേ ഉള്ളൂ. അത് പിന്നെ ഞങ്ങൾ വേറെ ഷെയ്ഡ് മാറ്റിയിട്ട് അത് ഫോൾഡ് ചെയ്യുന്നു.
അത് യൂണിവേഴ്സിറ്റി സ്റ്റുഡന്റ് ഏത് കോളേജ്? അപ്പൊ അതിന് തെർമൽ സ്റ്റെബിലിറ്റി നോക്കിയിട്ടുണ്ടോ? അത് ഉണ്ടാക്കിയ കോമ്പോസിറ്റ്
പോളിയാനിലിന്റെ ടി ജി എടുത്തിട്ടുണ്ടായിരുന്നു.
ഉണ്ടാക്കി കോമ്പോസിറ്റ്
ഇല്ല, അതിന്റെ എടുത്തിട്ടില്ല. അത് പല ആപ്ലിക്കേഷൻസ് ചെയ്തുകൊണ്ടിരിക്കുകയാണ്.
ചിലപ്പോൾ അത് ചെയ്തല്ലേ ഈ മെക്കാനിക്കൽ പ്രോപ്പർട്ടീസ് ഒക്കെ ചെയ്തു വന്നത്. അത് ഇവിടെ കട്ട് ചെയ്തിട്ട്
ഇത് ഫർദർ നമുക്ക് ജെല്ലാക്കി മാറ്റാൻ പറ്റും. So ഞങ്ങൾ
ജെല്ലാണോ? ജെല്ലാണോ?
അല്ല, ഇതിപ്പോ ഫിലിമാണ്.
ഫിലിമിനെ
നമുക്ക്
കട്ട്
മാറ്റാൻ
ചെയ്തിട്ട്
പറ്റും.
മെറ്റോഡിക് ഉപയോഗിച്ചിട്ട് അത് ഷീറ്റുകളാക്കുക. എന്നിട്ട് കറക്റ്റ് ഇൻസ്ട്രുമെന്റിൽ കട്ട് ചെയ്തിട്ട് സ്ട്രെസ് സ്ട്രെയിൻ ഇൻസ്മോഡസ് ഒക്കെ വെച്ചിട്ട് മെക്കാനിക്കൽ പ്രോപ്പർട്ടീസ് സ്റ്റഡി ചെയ്യാം. അപ്പൊ അതൊക്കെ നല്ല വ്യവസ്ഥ കേട്ടോ. ഞാൻ ചെയ്തിട്ടുണ്ട് മന്ത്രി.
The next presentation is by Shweta B. Nair, College of Agriculture, Vellanikara.
So, okay, some PL, what okra, motion sesquilent is always the one of the favorite vegetable of Indians, especially Keralites. But many constraints faced by those who are producing this okra. Many pests and diseases. So I'm here with Bindi yellow mosaic disease, one of the major constraints faced by the farmers who are growing the craft plants. So Bindi yellow mosaic disease, which is a viral disease, we can see the symptoms like there is a vein clearing is a major symptom. Will be also affecting the fruits also. So I am here with presenting the topic seed association and vertical transmission potential of Bindi vein mosaic virus in okra. I know most of you are not from this biotechnology or plant or agriculture background, but our theme of presentation is also very broad and also let's have no boundaries. I hope you will be open enough to explore this topic. This is basically from a plant pathology topic. So this is propagated through seed. So I am studying here whether this disease can be transmitted through the seed in next project. So this study I have divided it mainly into 3 parts. Like first is the collection of seed from the infected plants. There I am ensuring that that plant is containing this disease through symptomatology, that is visually and by the molecular confirmation using PCR. As it is viral disease, as we have done for COVID and all, we have confirmed it using the molecular technique, like PCR technique, polymerase chain reaction technique, serological confirmation. It is this antibody interaction text, mainly ELISA, enzyme linked immunosorbent and I have used. Then, after confirming that plant is containing viral disease, I have seeds for that, and that seeds I have evaluated for the seed association of the virus. Is whether this virus is present on the seed, any part of the seed. Then, when for the actual study, like study, where the information of the seed transmission, whether it is transmitted to the next project is evaluated. First, I have done a purpose sampling survey in different locations of Kerala and collected seeds from mainly 3 different varieties: Anjida, Shaubaha, and Amkombat. Anjida is one of the most cultivated variety in Kerala, and Shaubaha is not from Kerala Agricultural University, but it was reported as a resistant for this virus. But during my study, I have observed that resistant breakdown is observed in this workshop variety. And which is a local variety. Around 56 were collected from the infected plants. From the infected plants, for confirming this plant is containing virus, I have taken seed leaves from that samples, extracted the virus DNA using stab method. It contains some 6 presentation and purification step, and after that, I have quantified this quantity of the virus, quality of the virus using nanodrop spectrophotometer, then serological confirmation ELISA, PCR, and in PCR I have used virus specific primer, then primer, which is Bergamo specific, belongs to the Bergamo virus family, and that specific primer I have used for PCR. The DNA, the viral DNA was sequenced using sequence and interpretation analysis was using FAST software. Thus, this leaf I have used leaf for the study detecting the viral DNA. I have extracted the DNA quantified for 30.4 nanograms per microliter of the DNA particle was present and sequenced it. This is ALOYSIUS gel electrophoresis image where the PCR product and brand, and it was the 50 base pair size. The required Amazon size this particular virus. And by last analysis of the sequence, I have found that this virus is having 96.27% relativity with the yellow mosaic virus isolate of segment DNA A. This serological detection result is this is an ELISA plate. This plate is an STM plate. In that plate, we are doing that ELISA. This color change you can see some are having yellow color. That is the result we are observing. We will observe using the ELISA plate reader. Using this change quantity we will be estimating, and from that I got the 20.7% difference between the Other method is TBAT or TBAT. As it is done on NC paper sheets, and also have got 37.7% increase in that sample of the head. Confirmed that this sample is containing virus, and seeds were collected from that. The fruits were dried, seeds were not, and seeds were collected. So some next part of the study was seed analyzed. The whole seed was analyzed, and also seed parts. Different parts of the seed got hidden, and germination tube was studied. For that, the seed was germinated in water, sterile water, and the parts of the seed were separated. During molecular assessment, the same as I described before, PCR extraction, PCR, analysis, all of that, and that have for 21 seeds have analyzed for four species joint and con. The presence of virus was detected on the seeds, maybe 20% of seeds. But it was failed to detect the low application of the virus that seed or germ present, because not all seeds are containing this virus. This is the all electrophoresis image. There are for image where the amplicon in the different parts of the analysis, amplicons obtained. This is possible control I have there. The seed transmission, the third part, evaluation of this transmission to the patient. This virus is basically insect transmitted virus transmitted by Babesia tarsi. So, ensuring a set of condition is very essential for studying whether seed transmission. I have ensured the set of condition for study, and the collected seeds on, and the study was that around 8 seeds have sown but on 154 were germinated. This germination percentage was actually approximately around 8%, 7 to 8% germination. Here you got only 54% germination. This is not from the infected plant. Indicate that there is some effect on the seed quality by the infection. And the presence of also symptoms were observed on the plants, also the amplicon that
can you please elaborate the future outlook of this project?
Yes, ma'am. Future like seed transition or coordinate measures all are implementing. So here we can